of this experiment is to dissect and identify the male and female parts of a complete flower. The materials required for performing this experiment are a complete flower. I'll be using hibiscus in this case. You can also use China rose or mustard, whichever flower is available easily. You need a needle or a forceps, a blade, a hand lens or a magnifying lens to see the parts of the flower clearly. Let us now do the experiment. Let us now observe the male and female parts of this flower. So this is a hibiscus flower. As you can see, yellow dots, these are the anthers, and this is the stamen, and in the center we have the female part, that is the pistil. We'll now dissect this flower so that we can see the parts individually. So the outermost whorl, this is the epicalyx. So I'll just gently remove this. So you can see the small little green leaf-like structures that are there. This is the epicalyx. Just inside the epicalyx, this is the calyx. A small cut here so that the parts can be removed easily with the help of this blade. Now these green leaves that you see, this is the calyx, which is made of sepals. So you can see the purpose is just protective. Means they protect the flower when it is a bud. So we'll remove this very gently. You have to be very careful because it's a very delicate part of a flower and we don't want it to break off. So we have removed the whorl of sepals, which is the calyx. I'm placing it here. So this was the epicalyx. Now we have the calyx. Now the beautiful red petals, these are the colored part of the flower. Purpose is to attract the insects for pollination. And again, very gently, very carefully, we'll be removing each one of these. So there are 5 petals in all in this flower. So it is actually the petals which are meant to attract the insects. That's why they have such beautiful colors and they have such beautiful fragrance also at times. So we are just removing the petals from the flower. So now this is the reproductive part of the flower. Means the male and the female reproductive parts both are present in this. Now we have to just you can see these yellow dots, these are the anthers, and if we pull it off, we can collect them, and if you break it and you observe it under the low magnification of a microscope, you can actually even see the pollen grains. So the pollen grains are there inside these yellow these are bilobe structures which are known as anthers. So very gently, very delicately, you can remove the anthers. And now inside this red structure that we have, now again this is something that has to be done very carefully. We'll just break it off from here, giving it a very gentle cut, and you can see the ovary here, the structure that has come out. This is the ovary. A gentle cut through the structure. Small part is still stuck. and here we have, this is the female part of the flower. You can see it so clearly here. This is the ovary, this is the style, this is the stigma. This is the male part of the flower. So the stalks are fused in this case, the filaments are fused, so we have the anthers on top. And if you dissect the ovary further, you can even see the ovules. But for that you need a magnifying glass, which I won't be able to show on camera, but yeah, you can definitely do it when you're doing this activity in the lab. So I've just given a gentle slit to the ovary and we have the ovules inside this. So this is what the male and the female reproductive parts of a flower look like. So the outermost whorl, epicalyx, calyx, corolla, and then we have the whorl of the male part, which is known as the androecium, and the whorl of the female part, which is the gynoecium. So this is how the structure of a flower is. So we can now conclude that the stamen is the male reproductive part of the flower. It has a long slender stalk, which is known as the filament, which holds the anther. Inside the anther, the male gametes are there, which are known as the pollen grains. And likewise, right in the center of the flower, we have the pistil, that is the female reproductive part of the flower, which has three parts: the stigma, the style, and the ovary. The stigma is sticky so that the pollen can stick on the surface for pollination. The style is hollow, and inside the ovary we have the ovules, which are the female gametes. So the conclusion drawn is the male part is the stamen, the pistil is the female part, which we have already discussed. Pollen grains, which are inside the anther, they are the male gametes, while ovules, which are inside the ovary, are the female gametes in case of plants.